Hello everyone, my name is Chris, and the topic of my project was to use microarray analysis in order to classify cancer gene expression data. So first off, a little bit of background. The cancer concerning my microarray data is non-Hodgkin's lymphoma. It is a cancer that affects a person's white blood cells. As we all know, cancer is a mutative process in the cells that can prove deadly. Um, since non-Hodgkin's lymphoma affects white blood cells, it also has the side effect of compromising a person's immune system. It makes up about 4% of cancers in the United States and is quite deadly with a death rate of 25%. There are two subtypes of lymphoma, diffuse, which tends to be the more aggressive kind, and follicular, which tends to be the slower growing variant. My project's task is to take the microarray data of lymphoma cancer samples and classify them as either diffuse or follicular. Since taking this class, I think we all have a general idea of what a microarray is. In essence, it's just a grid that can hold the genetic material of various biological samples. Each slot or column in a microarray can be organized accordingly to correlate with a certain DNA probe. Um, the strength of the probes binding to the biological samples gives us information about the samples. For example, if a probe for a certain gene binds to a sample, we will observe its fluorescence, and we can conclude that this biological sample can be associated with a certain gene. So my project's main task is a binary classification problem, as mentioned. This means I need to classify my lymphoma cancer samples as either diffuse or follicular. To do so, I've chosen to implement the KMeans clustering algorithm. By being able to classify samples successfully, it allows scientists and medical professionals to have a method that aids early screening. In addition, through the classification process, as you will see, we can also identify the most relevant genes from the dataset. With this information, it can provide a basis for targeted gene therapy regarding this cancer. So here's a snapshot of the microarray dataset. It's publicly available online, and you can find the link to it in the references attached to this presentation if you'd like to explore it. So as you can, this dataset has 77 samples or rows and 7,129 genetic probes or columns. So the genetic probes or columns are also referenced as features. And it's quite common for microarrays to have a much higher number of features than the number of samples. This leads to the problem called the curse of dimensionality. In high dimensional space, it means that for each sample, there are more values to consider when doing calculations. This can get very computationally expensive for more intensive algorithms, and also the data points in the high dimensional space are like finding a needle in a haystack. Without sufficient samples, an algorithm or model cannot gain any valuable information. In such a case, a model may memorize the unique high dimensional values of these samples, but when it is tested against unseen data, the model does not perform well. This is also known as overfitting. So the goal is to have a model that can perform well on the data we feed it, and also perform well on unseen data. So to counter high-dimensional microarray data, I utilize a dimensionality reduction tactic called principal component analysis, or PCA for short. Um, Simply put, we have our data in n dimensions. We find these new axes such that when we project the data onto these axes, the axis captures the maximal variance of the data. These axes are also known as the principal components. So through PCA, I reduced my data from 7,129 features to 2. So the number 2 was chosen so that we can visually observe the data points in 2D. The general process of PCA is that we first subtract the mean from the data within each column, calculate the covariance matrix, apply the we apply certain matrix operations to find the eigenvectors and the eigenvalues, and the corresponding eigenvector with the highest eigenvalue corresponds to principal component 1. And principal component 1 is the axis that captures the most variance of the data. So here I took the top two principal components and projected the samples onto them. We see that principal component 1 in blue captures the most variance, followed by principal component 2 in pink. Capturing variance of the data just means that it's able to capture information from the data set. Since PC2 is more scrunched together, it's hard to extract any useful information compared to how the data in principal component 1 is more spread out. So we have more information about the data set, which is ideal. So normalization is also an important component regarding microarray analysis. Normalizing the data values just means to make the data on the same scale for all features. And after normalization, the values will range from 0 to 1. This is important because it allows consistency among features and it gives better interpretability of the data. So before, the values range from some thousands to 16,000, but when we normalize here, you can see that they range now from 0 to 1. So then we apply principal component analysis onto this normalized data set, and the values of the samples in the principal component space, they will no longer be from 0 to 1 because of the projection, but they will remain on the same scale, more or less. And this is what it looks like here. So on the left is the normalized dimensionality reduced data. Like I said, I chose two principal components so that we could visualize the data points in 2D. Since we had originally like 7,129 genetic probes or features or columns, we cannot visualize a 7,000 dimensional plot. And on the right is a scatter plot for the data with the ground truth labels. So these labels are part of the public data set, and they were assigned by the original researchers. So these labels will be used to compare our K-means clustering labels to assess our model's accuracy. Also, we will set aside 8 samples to test our K-means model in order to evaluate its performance on unseen data. So K-means clustering, how does it work? K-means aims to cluster data points into local regions, assuming that neighboring points are the same type. The first step is to initialize k-random centroids. Since this dataset only has 2 classes, I set k to 2. For other datasets, maybe we do not have the labels or we do not understand the data, then we can utilize the elbow plot to test k, the value of k, to find the ideal number of clusters to use for that data. But since this dataset, I know there are only 2 classes, we set k to 2. So then next, we calculate the distance of each sample to each centroid. Now that sample or data point belongs to the closer centroid. So now we do that for all the samples, and we now have 2 clusters. Calculate new centroids from these clusters, and we repeat until the centroids converge. K-means assumes that the nature of the data can be grouped into circular regions. This may work to some degree, but it is important to know that some data may not be so easily classified by grouping into circular regions. So after running KMeans with 2 centroids, here are my results. On the left are the centroids and clusters obtained from KMeans. We compare the predicted classifications to the ground truth labels that were annotated by the original researchers, and I was able to get a 76.81% training accuracy. So for a baseline model, I think that's quite good. And on the right, we observe the 8 samples that we left out, and we test our model on these 8 samples. They all happen to be the diffuse class, and we were able to get 50% accuracy. So if you look at the plot on the right, it makes sense because there seems to be like 4 plots closer to the diffuse centroid, and the other 4 dots belong to follicular centroid on the left side. So it makes sense why we would get 50%. So I was wondering, could I do better with the K-means model? Um, Outliers are something to consider in microarray or any dataset for that matter. So I utilized the median absolute deviation method, the details of which are in my write-up for your reference. Put simply, we look at the original data before K-means. Using the ground truth labels, we calculate the centroids and each sample's distance to their respective centroid. So the centroids you see here in this plot are not the K-means centroids. Since we know the labels of the original data, we can calculate the centroids, and then we identify the median distance for each sample to their respective centroid. Then for any sample that diverges too far away from the median distance, we classify them as outliers. So in my experiment, I was able to identify 2 outliers shown by the big light blue dots. Removing these outliers and reapplying K-means to the dataset, we see a slight improvement from 76.81% to 79.1%. However, the test accuracy remained the same. And that's because the removing 2 outliers was not sufficient to move the original K-means centroid. So we were still getting the same results in the test set. So some things to consider. Remember that this dataset only has 77 samples. Perhaps K-means would perform better if there were more samples to help converge the centroids to their optimal locations. If so, we would see an increased accuracy on the test data. And also, by observing the position of the follicular and diffuse samples, it appears that clustering cannot create optimal classification boundaries. So out of curiosity, I went ahead and implemented the support vector machine algorithm, or SVM for short. The details of which are out of scope for this presentation, but basically SVM is a more flexible algorithm in its ability to classify samples. While K-means is restricted to circular regions, SVM can draw non-linear decision boundaries. So just by looking at the lymphoma cancer dataset, we can imagine a non-linear boundary as shown in green on the bottom plot. So this is much more detailed than the circular regionings of K-means clustering. So with SVM, I was able to get a much improved 95% training accuracy, and test accuracy improved from 50% to 87.5%. So this confirms that the nature of the data and the algorithm chosen can influence your performance. To conclude, I show that K-means performs adequately in classifying lymphoma cancer. With this knowledge, scientists can take appropriate screening treatments, especially in the early cancer stages, to ensure higher chance of survival. Through principal component analysis, we can identify the features or genes that contain the most information from the dataset. This allows identification of target genes and can help focus research efforts towards these specific genes. And overall, this can help save scientists and researchers time and money. That concludes my presentation. Thank you very much.